If you're a scientist with an idea that want to found a company, do it, but prepare for the journey. If you're working in upstream bioprocessing, and especially in the areas of fermentation, and you are working in shake flasks, go and read up on what tools and options are out there. For example, come to our company, Scientific Bioprocessing, and take a look at the material that's completely for free, and you can just run better shake flasks as of tomorrow.
Hey, smart biotech scientists, welcome back. We are in the middle of a conversation about the use of shake flasks and how you could potentially transform a shake flask into a bioreactor. And my today's guest is Jens Bayer, who is a deep tech entrepreneur and experienced commercial leader, and he currently serves as the Chief Commercial Officer at Scientific Bioprocessing. He also co-founded Aquilo Biolabs, and under his leadership, the company experienced rapid growth and a successful exit through acquisition by Scientific Bioprocessing. If you haven't listened to part 1, please go back, and today we are diving into the use of sensors and how this will enable you to augment your shake flask system. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brohlman, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. You are working on ways to transform shake flasks into bioreactors. So you have now these DOTS platforms. So can you tell us what this platform is and how this platform enables exactly that?
So maybe to explain why we came up with this DOTS platform. So the idea was simple. We saw the advantages of a shake flask, simple to use, low cost, low footprint. Everybody's using it, but we saw what was kind of missing in order to turn it into bioreactors, right? Like predictable, controllable data, and just that it's a little bit more engineerable. It's more controllable. And we felt like, is there no way to kind of combine this? So maintain the advantages of the shake flask, but kind of add to it what people like from bioreactor or Amber. And that's how we came up with the DOTS platform. The DOTS platform, the idea of it is very simple. Turn your shake flask into a smart bioreactor. That's kind of the vision of it. It's telling people you have a shaker, you have your flask, and now we're giving you tech. You add the tech to this setup, and you're basically getting bioreactor-like capabilities in your flasks. And the way we do it is that there are 4 components in the platform. We have a sensor that you put on your normal shaker below the shake flask, and it's a so-called multi-parameter sensor. So this is a very powerful sensor that can monitor backscatter at different wavelengths. So for example, like this, you get non-invasively growth curves for your culture. So you can literally look at the growth curve and determine, now my culture is here, now I want to sample, now I want to take it out, now it's stationary phase. You can compare, we have customers that use up to 100 different shake flasks and compare the different batches and runs and strains against each other. You also have fluorescence measurement on this sensor. So you get, for example, real-time data about your protein production, if it's GFP tagged as an example, or if you're running promoter studies and you're using something like photo-switch proteins. So you have a lot of data coming already from the sensor. In addition to this, we have the second component that are sensor pills. It's a new generation of chemosensors that we have developed, and people are interested in more than just biomass and fluorescence. They want pH, DO, glucose, substrates. So there is no non-invasive tool that can measure this at the moment. That's why we went down the chemosensor route. And imagine a sensor pill really like a small paracetamol pill, and it comes as sterile use. It can detect a certain analyte, and you just throw it in your flask. And then the pill circulates with the liquid in the flask, and every time the pill passes the sensor, we read it out, and by that we can now understand, for example, what's the DO, what's the pH, what are other analytes doing in this cultivation. And our vision really is to build a broad portfolio so that the scientist just drops in the pills that they are interested in, the data, and then they get it exactly. And you can always fine-tune it for every experiment. Maybe sometimes you're not interested in a parameter at all, so you don't put any pill. If you want to have certain, you drop in the relevant pills. That's the second component. So you can see we have very focused on data, giving you a lot of data, but the data you want in an automated way without sampling. The third component is a feeding system that you put on top, and now you can run in your shake flask things like fed batch. So imagine you monitor your DO, and you want to say every time the DO spikes, or we have a set, I want you to feed. So something like this that you do in a bioreactor quite often, you can now do on a shake flask level. You could also control your pH like this. So you can see now there's a control option that we're adding to the shake flask. And the fourth option really is software, because I've talked a lot about tech here. And in order to control it, in order to plan your experiments, and of course, also to analyze the data, you need a good software. And that's the fourth component. And that's really what the DOTS platform is, a way to turn your shake flask into a smart bioreactor.
And how easy is the use of the DOTS platform? Because as you have highlighted before, we have quite a diverse industry. We have very different media. We have different organisms. We have a lot of different factors. How much work do I need to put in order to make this system work and to get something meaningful out of the data we are generating?
So at the moment, I want to highlight the DOTS platform is available commercially for microbial processes, where we've tested it for yeast, for bacteria, for filamentous organisms. So there it works. For the cell culture space, if you're talking about show cultivations, for example, that's where we right now are working on pilot tests to also show that it works there. But in a setup, it's very easy. So the sensor, you just install this once on your tray, and afterwards all you need to do is put the flask on. So you don't need to do something with the sensor all the time. It's literally equipping your shaker tray once. And then you just put your shake flask in and on like you're used to. The pills is, I would say, whenever you put your media and your cells in your flask, you just decide, do I want to put a pill in as well, and which one? So it's adding one step, but it's as easy as opening a sterile package, dropping in the pill, and you're done. And from a feeding point of view, the only thing you need to do there is it's a module that comes on top, and we have a cartridge that is single use and empty. So you can, under sterile heat, you fill it with any liquid that you want, any mix that you want. You put it together with our feeding system, put it on top, and you're done. It's also single use. So I would say you can put the DOTS platform up within 10 minutes, 10 to 15 minutes max per experiment. 15 would be on the slow side. If you compare this to how long it takes to set up one reactor, it's a long time, and we're not talking even about cleaning after the experiment, right? Because you throw the pill, you put the shake flask in an autoclave, and you're restarting, and whoever has cleaned a bioreactor knows what I'm talking about, what awaits you once you're done with your experiments there.
Are there any cell types or any media that would not work with your system, or where you have observed some certain limitations?
Sensing in bioprocessing is very complex, so we will need to break it down now by parameter. So, for example, we use backscatter in order to measure biomass. And if you, for example, have a media that has a lot of particles in that, so let's say you use a very cheap substrate, but it comes, with wood pellets and stuff like this. We've seen all kinds of stuff, really. And let's say at the beginning of the cultivation, you have big pellets, big chunks. And during the fermentation, they start to break down into smaller and smaller pieces. These particles, of course, also scatter. And of course, if you have a couple of large chunks in the beginning and then many small ones, that's a very different signal. So this can have an interference. For the pills, for example, it's important that the pill rotates together with the liquid. So if you had a very, very thick media, maybe talk more towards juice or smoothies or something like this, then of course the pill cannot rotate like this predictably, and then we can't read it out. So I would say in our company, we have experts working on this. So everybody who is interested will go kind of to an interview with us where we look at the bioprocess details and we can make a recommendation. And we have a very big database around what works and what not. So usually we can quite quickly tell the customer if this looks good or not.
Now, this all sounds very exciting. Where do you see this going in terms of technology? Or you were in this process of augmenting the bioreactor. Let's assume we're having the same conversation 5 years from now. What will have happened?
That's a good question. I wish I know if I would place bets on it. But if I had to guess, educated guess here is, we see AI everywhere right now. And I think it's pretty clear that we need data, especially fermentation data, as cheap as we can get it. I think the shake flask can be a way. I see a world where the DOTS platform and other technologies will deliver this fermentation data in real time, and where really AI takes a little bit over of the guesswork that's partially going on, especially in the screening and the selection and the optimization space. There's a lot of DOE also involved in this. So I definitely think that AI is going to be a topic, but I also think it's going to be more boring topics, right? AI is a pretty hype topic, but I think it's as simple as making sure that you get comparable oxygen transfer rates in shake flasks than you get in bioreactors. So you can be limited there, right? People use dendephils or they shake faster, but like in bioreactors with active gassing and stirring, you just get more oxygen in. And I know for a fact that there are a couple of people right now in the space really working on innovations. I've seen them myself. Some of them have been presented on conferences. So if we really take this part of the equation and add it to the shake flask, then I think we really can turn shake flasks into bioreactors. And I think the third lever is there will be more data. I'm thinking glucose, I'm thinking viable cell density, especially for the cell culture space. So there will be innovation on what data can you actually get from everything. And I hope that then the shake flask can help, especially in the synthetic biology space, because many of the challenges that we face as society at the moment, I think they require synthetic biology products and revolutions, and I think we need to lower costs and help more products get to the market, especially in that space.
Having technology is great. Having the right tools to generate amazing data is great. And also, if you're able to select the right tools that simplify your life. But there's another thing we need to talk about, because you need also to design the right experiments. I mean, you need to ask the right questions and then also make sure you're getting the data to answer the questions you have. Can you give us some strategies to define successful shake flask experiments?
My number one tip is read up about shake flasks. Similar to somebody gave you the task, hey, you need to learn how this bioreactor works, right? You would study the manual, talk to the manufacturer, read about bioreactors, and I recommend the same for shake flasks. So we, for example, in our company, Scientific Bioprocessing, we have a blog where you can read about the most common challenges, but also I want to highlight, go to shaker manufacturers like Inforce or Kuna. They also have great libraries. Just build knowledge around it so you know what you do. I would definitely recommend you to take a look at oxygen, especially when you're working with E. coli or other bacteria that grow quite fast, because that's the number one limiting factor that's probably also preventing predictable scale up. So usually people make their flask too full. So go for something like 10% and not above. I've seen up to 50%. And if you combine high filling volumes with very low RPM, you are guaranteed to almost be oxygen limited. So low filling volumes, let's say you put in a 250 milliliter flask, you put 25 milliliters of media. You should aim for shaking speeds that are high, 250 RPM plus. If you can, go up to 300, 350. If you look at Professor Büchs, again, I've mentioned him a lot today, they shake at 3 to 4% filling volume with 350 RPM on 50 millimeters throw, right? These are extreme conditions. And if you look at these shakers, you just have liquid stuck to the wall. But that's how you create the biggest surface area. And that's how you generate the oxygen transfer rates that are needed so that your organisms are not limited. That's one thing, but you can do all of that. If you then put it close to shake flask with aluminum foil, then you're screwed anyways, right? I would say that's the most common mistake I see. Put aluminum foil on your mouth, David, and try to breathe, and I think you will understand how your E. coli cells feel. So don't use aluminum foil. Go for cotton plugs, go for membranes, go for something else. And then the last one is just use more technology, right? If you want to plan your experiments better, I mean, every scientist knows that when you're operating blind, science gets way more difficult, right? The more technology, the more data you get and you actually see what you're doing, the faster you will have results and the faster you will need less experiments. So I'm always surprised how little people know that there are now sensors, so you can automatically generate growth curves, that you can look at the DO in shake flask, that you can run fat batch in shake flask. And when we meet people on conferences, they're like, I didn't know this existed. I've never heard of this. And I think it's really an awareness problem. So just go out and Google a little bit around shake flask and you will be surprised how much tech is out there that will help you experiment.
Fantastic. Thank you for making it so practical, Jens. Now in this final part of our conversation, I'd like to circle back to your career and especially to your entrepreneurial endeavors, because you had this opportunity to found a company, to grow it, and actually even experiment and exit. So can you tell us a bit what were the most valuable lessons along this exciting journey?
Let's explain the journey and then come to the learnings that I had. I studied actually first biotechnology, like I said, in Aachen, and then I studied bio-entrepreneurship in Stockholm. That's actually a program where people with a scientific background kind of do a mini MBA in order to learn the business side of things. And when I was done with this program, my two former friends, or not former friends, they're still friends, but like we studied together, and they said, let's found a company together. We have an idea, you run the business side. So we then did this together with one guy, Daniel from Stockholm. So we were four of us. And the journey in stages was like this. We first exist scholarship in Germany. It's a program that helps you in the first year when you have an idea, but you're not investor ready. You need to kind of build your business plan, your pitch, proof of concept, take care of IP. You need time and a little bit of money. That's what the scholarship did. And the German government luckily rewarded us. We worked through that and then really got investor ready and closed a seed round. So seed investment round. Our biggest investor was actually Dr. Jürgen Schumacher. He's a business angel and co-founder of QIAGEN. Really, really helpful on this journey, and we owe him a lot, in my opinion. And then from there, we really scaled the company from 4 boys with an idea to 15 people, customers in 25 countries, 3 products, strategic partnerships with Infos, the shake and bioreactor manufacturer, and then sold it to SBI in 2021. And during this time, I was one of the co-founders, especially with a focus on commercial topics. And I would say my main learnings are it's a very tough journey. So whoever thinks that entrepreneurship, there are often these fancy stories out there, and you could tell this story in a fancy way, but the reality is you need a lot of resilience. You face a new challenge every day, and there are so many times where we could have given up, but we didn't. So I think you need to be prepared for this journey. It's really like climbing Mount Everest. And you need flexibility. If you think as a founder, I just take care of topic A, that's not how it's going to be, because you're going to be suddenly we have a production issue, or there's a problem with the contract. So you need to constantly adapt to what the challenge is today. And no, there's nobody else, right? You cannot say, oh, but this is department X. There is no department X that takes care of this. It's you, the founders. So you really need to be flexible. And in the end, it all comes back to the people. So if you go on this journey alone, or if you don't have a great team, you will not make it. So wherever you work, you need great people and a good team around you. So that, I think, were the key learnings that I had.
What advice would you give scientists who are listening today who are on the fence about starting their own startup?
Do it. I loved it. I would always do it again. What impressed me was that I'm not a big quote guy, but one thing stuck to me that somebody said, and it's Steve Jobs who said, in innovation, ideas are only part of the equation. Execution is just equally important. And that's true. I think the scientist, if you have an idea right now and you're thinking, we need to found a company, it's very easy to think, I've done all the work up here. Now we just need to add a business person who sells this stuff, right? That's very often how scientists think. And the problem is that's then the later execution of the idea. And I don't blame them. It is easy to think like this. But what the execution of this idea to a real product that then sells to a company that works and grows, that is equally a challenge as coming up from an idea to the actual technology that you may want to commercialize. So I would say to the scientist, team up with somebody on the business side so that you can focus on your core capabilities and strengths, because often you don't want to take care of all these topics that are part of execution. You want to focus on the technology. So team up with somebody. And then also make sure that, split the company evenly, in my opinion. So if you're talking shares, it's a big cake. And I understand that people say, hey, I've invested 5 years of research and now this business person comes along. Why should they own the same part of it? But I was impressed back then of my co-founders that they said, no, we split this evenly, because later on, we will need you equally much as what we did so far. And this really, from the first moment, sets the tone of we are one team. It doesn't matter. Everybody is in the same way motivated. And I think that's the advice that I would give to everybody. And the last one is stealth mode. I think that's one of the biggest issues. It's like so many people think, I have this idea. I should not be talking. Maybe somebody steals it, right? But if you're thinking about it, especially as a scientist, the probability that you meet a person that knows this better than you, that right now has the time and the money and the motivation to take this idea and turn it into something better than you can, it's so small, but yet so many people stay in the stealth mode. And that's also something we did. Go out there, talk to investors, talk to competition, talk to your users, but go out there and learn. I think you will be surprised how much this helps compared to the risk that you're taking with it.
Exciting. We have covered a lot of ground, Jens, today. What is the most important takeaway from our conversation?
If you're a scientist with an idea that want to found a company, do it, but prepare for the journey. If you're working in upstream bioprocessing, and especially in the areas of fermentation, and you are working in shake flasks, go and read up on what tools and options are out there. For example, come to our company, Scientific Bioprocessing, and take a look at the material that's completely for free, and you can just run better shake flasks as of tomorrow. I think that's the thing. Like, the knowledge is there. Now you're aware of that. You should be treating it like a bioreactor. Inform yourself and just do it. And then I think we are in a very exciting age where really hardware, chemistry, software, innovation comes together to improve upstream bioprocessing. And I hope that we really, over the next 5 to 10 years, will see huge improvements in success rates, faster times to market, and just more innovation coming out to solve some of the biggest challenges that we have. That's what we are dedicated to, and that's why I'm standing up every day and being committed to SBI and the journey we're on.
Jens, where can people connect with you?
On my LinkedIn, if it's for me personal, so for example, if somebody would like to have some advice around founding a company, then Jens Bayer like the pharma company. I want to highlight I'm not related, it's just a coincidence. So on LinkedIn, and otherwise, if you are interested in the technology, the DOTS platform, learning more about shake flasks, and maybe even trying out some sensors for your own bioprocesses, then I would recommend you to go to scientificbio.com. That's the webpage of our company. Take a look around, and you will find options there to get in contact with us.
Smart biotech scientists, I will leave all the links down in the show notes, so please reach out to Jens, talk about entrepreneurship, and also check out the DOTS platform. And thank you so much, Jens, for sharing all your insights, your experience, and for being on the show today.
Thank you for the invitation, David. And I want to highlight, I heard from this podcast first from a customer who talked about it. So I think you're onto something here. People love you in the space, especially the people that work every day in science around it. So continue the great work, and thanks for putting the podcast up.
Oh, that's awesome. Thank you so much for this feedback. Thank you very much for tuning in today. What do you think about today's episode with Jens Bayer? Please tell us by leaving a review. And if you still have questions about whether you should be using shake flasks or whether you should be using a more sophisticated bioreactor system, or just any question about process development, I want to help you get clarity. Just book a free 30-minute assessment with me using the link down in the show notes, and I'm looking forward to seeing you on the call. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.